Hello everyone, my name is Siti Salma with my partner Muhammad Farid Rizwan. We'll be presenting on our assignment Analytical Biochemistry for our assessment today.
Electrophoresis is a laboratory technique used to separate biological molecules like proteins or DNA using an electric current. One-dimensional electrophoresis separate molecules in one direction based on one property, usually their size. Two-dimensional electrophoresis separate molecules in two steps using two different properties. First, proteins are separated by their electrical charge and then by their size. This results in proteins appearing as spots on the gel.
Is electrophoresis. Electrophoresis is a laboratory technique used to separate biological molecules such as DNA, RNA, or protein based on their size, charge, or shape by applying an electric field. The molecules are placed in a gel, and when electricity is applied, charged molecule moves through the gel. Small or more highly charged molecules move faster and further than larger ones. Electrophoresis one-dimensional. One-dimensional electrophoresis separate molecules based on one property only. Only one gel is used in 1D electrophoresis, that is polyacrylamide gel. It is a protein that separates based on molecular weight. SDS denature protein and give them a uniform negative charge. Smaller protein move faster through the gel pore. The separation occurs only in one direction. Two different gels are used in 2D electrophoresis to achieve higher resolution separation. First dimension gel immobilized pH gradient strip contain a fixed pH gradient used for isoelectric focusing. Proteins separated based on their isoelectric point. Proteins stop moving when net charge becomes zero. Second dimension gel, polyacrylamide gel, SDS pH, the same type of gel used as 1D electrophoresis that separate protein based on molecular weight. Produce distinct protein spot instead of bands.
Two-dimensional electrophoresis. Two-dimensional electrophoresis is a powerful laboratory technique used to separate complex mixture of proteins. It separates proteins in two steps, each based on a different property. 1. Isoelectric point. 2. Molecular weight. Because of this double separation, 2D electrophoresis provide very high resolution and can separate thousands of proteins in a single gel.
Difference between 1D gel electrophoresis and 2D gel electrophoresis. 1D gel electrophoresis is a process of protein separation based on their molecular weight utilizing polyacrylamide gel electrophoresis, whereby 2D gel electrophoresis segregate protein based on their protein molecular weight and isoelectric point. 1D gel electrophoresis is a protein separation based on molecular weight only. 2D gel electrophoresis is protein separation based on molecular weight and isoelectric point of the protein. 1D gel electrophoresis has low resolution, whereby 2D gel electrophoresis has high resolution. 1D gel electrophoresis not so costly, but 2D gel electrophoresis is very costly.
Principle of 2D electrophoresis. Protein differs in electric charge, size, molecular weight. 2D electrophoresis uses first dimensional separation by isoelectric focusing. Second dimension separation by SDS pH. Each protein finally appears as a separate spot on the gel. The electrophoresis apparatus is the equipment used to separate charged molecules under the influence of an electric field. Its main components are explained below. 1. Power supply. It provides direct current required for electrophoresis, allow controls of voltage, currents, and time, ensure a constant electric field during the run. 2. Electrophoresis tank, buffer chamber, usually made of plastic or glass, holds the buffer solution and the gel, contains electrodes at both end, anode and cathode, prevents evaporation and contamination. 3. Electrode, made of platinum or stainless steel. Anode, positive attract negatively charged molecules, while cathodes attract positively charged molecules, connected to the power supply. 4. Supporting medium, gel matrix, act as molecular sieve for separation, commonly used gel, agarose gels or polyacrylamide gel. Gel concentration affects resolution. 5. Buffer solution, maintains constant pH, conduct electric current, prevent overheating. Common buffer are TAE, TBE, and trisglycine. 6. Samples application system, wells or slot formed in the gel using a comb, used to load sample mixed with loading dye, ensure uniform sample entry into the gel. 7. Gel casting tray and comb. Tray holds the gel during casting and running. Comb creates uniform wells for sample loading. 8. Cooling system, used in high voltage system, prevents overheating and bend distortion. 9. Detection system, used after electrophoresis to visualize separate molecules, includes staining solutions and UV transilluminator. Process of electrophoresis. Electrophoresis is a laboratory method used to separate DNA, RNA, or protein based on their size and electric charge. 1. Prepare the gel. A gel of agarose or polyacrylamide is made and placed in a tank filled with buffer solution. Load the sample. The sample, DNA, RNA, or protein is put into small holes in the gel. Apply electricity. When electric current is switched on, the molecule starts to move through the gel. Movement of molecules. Negatively charged molecules move towards the positive electrode. Smaller molecules move faster through the gel than larger one. Separation. As they move, the gel acts like a sieve and separates the molecules based on size. Stop the run. After some times, the current is turned off once the molecules are separated. Visualization. The gel is stained so the separated band can be seen. Electrophoresis separate molecules by making them move through a gel using electricity, where a small one move faster than big one. The advantages and disadvantages of 1D and 2D electrophoresis. The advantages are stable chemically close-linked gel, sharp bend, good for separation of low molecular weight fragment, stable chemically cross-linked gel, while disadvantages is toxic monomers. Gels are tedious to prepare and often leak, and need new gel for each experiment. Analysis of protein purity using electrophoresis. A biotechnology laboratory produced a recombinant protein for research use. Before using it in experiments, the scientists needed to check the purity of the protein to ensure that no unwanted proteins were present. The objective to determine the purity and approximate molecular weight of the isolated protein using SDS page 1D electrophoresis. The result, the gel showed one clear sharp band at approximately 50 kilodalton. No additional bands were visible.
Second case study, disease biomarker identification using 2D electrophoresis. Many diseases cause changes in the types and amounts of proteins produced in cells. Studying these changes helps in early diagnosis and treatment. 2D electrophoresis is used to compare protein profile of healthy tissue and diseased tissue to identify disease-specific biomarker. First dimension, IEF, proteins are separated based on their isoelectric point. Second dimension, SDS page, proteins are further separated based on molecular weight. This two-step separation gives high resolution. Difference in protein spot, appearance, disappearance, and intensity changes are observed between healthy and diseased sample. Proteins that show altered expression can act as biomarker, helping in early disease detection and understanding disease mechanism.
Analysis of protein purity using electrophoresis. A biotechnology laboratory produced a recombinant protein for research use. Before using it in experiments, the scientists needed to check the purity of the protein to ensure that no unwanted proteins were present. The objective to determine the purity and approximate molecular weight of the isolated protein using SDS page 1D electrophoresis. The result, the gel showed one clear sharp band at approximately 50 kilodalton. No additional bands were visible. What is electrophoresis? What factor influence protein separation in electrophoresis?
What is 1D electrophoresis? What is the role of SDS in one-dimensional electrophoresis? Why do smaller proteins migrate faster in SDS page?
What is the limitation of 2D electrophoresis?
How can electrophoresis help in disease diagnosis? What improvement can be made to overcome the limitation of 2D electrophoresis? In conclusion, 1D and 2D electrophoresis is important in analytical biochemistry for separating proteins. 1D electrophoresis separate in proteins. One-dimension electrophoresis provide a simple way to analyze protein size. La maquinaria Intel.